Welcome back. In this video, we're going to talk a little bit about the cell cycle. Now, when I say cell cycle, a lot of you are thinking mitosis, but that's not the cell cycle. The cell cycle is a phase known as interphase for about 9% of the time, and only 5% of the cycle mitosis. Okay, so you need to change your thinking. It's in interphase, it's in this that the cell is going to grow. It's going to copy all of its contents except the nucleus. Okay. In mitosis, the nucleus gets copied. Everything else has to get copied in interphase. And in interphase is when the cell is going to prepare for mitosis. If we didn't have interphase, then a cell would not be successful in going through mitosis. So we need to talk about what's happening in interphase before we can talk about mitosis. Okay. It's only after we've done interphase and we have a final check the cell can undergo mitosis. And then after mitosis, the cell undergoes a process known as cytokinesis. Cyto meaning cell, kinesis, to cut. So it results in two daughter cells. And at that point in time, this is showing the two daughter cells, the cell would be ready to enter interphase again in what's known as the G1 phase. However, there are certain cells that don't go through interphase in the G1, and they would enter a resting state known as G0, where cells perform their functions, they never divide. And we'll learn about some of these cells later on in the course, because this is like what happens to your red blood cells. All right, so let's learn about the phases of the cell cycle. This interphase typically lasts around 24 hours for most of the cells in our body. And as I said, this is usually around 95% of the cell cycle. So we're looking almost a whole day, okay? And we're gonna have cell growth as well as a lot of metabolic activity. So let's look at the three different parts of interphase. The first part is known as the G1 phase. And in this part, we are going to be duplicating a lot of the organelles. And as you can see from looking at the drawing, this is almost half of interphase. The cell is going to grow a lot in size here. The cell doesn't grow in size, then it's not going to have a successful mitosis because there's not going to be enough cell to divide. The G1 phase is also where the cell does the work of the cell. All that transcription, that translation, you know about all those enzymes come to do the work. So all those chemical reactions are going to be happening. So everything we talked about, all the functions of those organelles, that's what's happening here in the G1 phase. After G1, it goes to the S phase, which stands for synthesis, but it's kind of a misnomer because what's being made here are not the organelles, but what's being made here is the DNA. So the chromosomes are duplicating, and yes, the centrosome. So there's like only one organelle duplicating here. Okay, but it's very important because we're going to need this for mitosis as well. From there, we move to the G2 phase. So in other words, okay, now we think we have duplicated everything. So let's get ready for mitosis. We're going to do a little bit of more protein synthesis, and we're going to have a check system. We're going to double check, is everything ready? Were there any errors made when we duplicated our chromosomes? Is all of our DNA okay? All right, if so, then we're going to be ready to move on to mitosis, okay? That cellular division. And it's only when we get to mitosis that the nucleus is going to reproduce. So notice, in the phases of the cell cycle, the organelles duplicate in the G1 phase. The chromosomes as well as the centrosome reproduce in the S phase, and the nucleus reproduces in mitosis. And at the end of mitosis, we will divide all the cell content in that cytokinesis, so we'll have two identical cells, both of which were identical to the parent cell that started the interphase. So when we duplicate the chromosomes or replicate the chromosomes, remember they're going to be homologous. They're going to have the same genes on each of them. We are going to go from a single chromosome or chromatid to having sister chromatids attached at that centromere in the middle of them. Okay. So if we were to look at cells in interphase in this one where so much is going on, the DNA is existing as chromatin, not as chromosomes. We have a very visible nucleolus because we've got to be able to make RNA. We've got to be able to make ribosomes so that the cell can do its work, make proteins. The nuclear envelope is intact, and the centrioles are going to be off to the side of the cell. If we look at photomicrographs, we can see cells in interphase are going to have a clearly defined nucleus. This one, you can clearly see a nuclear membrane as well in this one. And you can also see nucleoli. So this is what a cell in interphase looks like. All right, and with that, we are done learning about the cell cycle, and we are ready to come back in our next section and learn about the steps of mitosis. So thank you so much for all your hard work, and I will see you shortly.